Good afternoon, everyone, and welcome to the Andel Institute Public Lecture Series, Assembly Instructions for Cancer. I'm Steve Ozing, a Philanthropy Director here at Van Andel Institute, and I'm glad you can join us virtually for a presentation today highlighting the Institute's groundbreaking work in cancer research. To find cures for cancer, we must understand the nuts and bolts of how the disease starts. Today, we will look into cancer's molecular roots and how this research is revealing the fundamental mechanisms cancer uses to hijack regulation of our genetic code. Today, we will hear from Dr. Evan Worden, Assistant Professor in the Department of Structural Biology here at Van Andel Institute. Dr. Worden earned his PhD in molecular biology from the University of California, Berkeley, and was a postdoctoral fellow at Johns Hopkins University. He joined the staff here at Van Andel Institute in 2021. He has earned numerous prestigious awards for his research, including a Damon Runyon Cancer Research Foundation postdoctoral fellowship, the Paul Ehrlich Award for postdoctoral research from Johns Hopkins, and the Nicholas Cosarelli Prize for Best PhD Thesis from University of California, Berkeley. In 2020, he was a finalist for the Damon Runyon Dale Fry Award. If you find yourself with questions during the presentation, please submit them via the chat function or hold on to them till the end of the presentation. We'll have a formal Q&A session as soon as Dr. Worden is done. So let's begin today's program. Please join me in welcoming Dr. Evan Worden.
Thanks so much, Steve, for that great introduction. Yeah, so today I have the really nice ability or honor to talk about some of my favorite topics, which is the epigenetic regulation of our cells and how that relates to development of cancer. So I'm sure many of you are familiar with IKEA, right? And IKEA has this famous design for their instructions. And this is a Poang chair from IKEA. And I actually own this specific chair as well. But IKEA has very, very clear instructions that most of the time allow you to very faithfully get to the final assembled piece of furniture. But if these instructions are not clear, so for instance, in this IKEA knockoff here called Paradox, if these instructions are not clear, then you're not going to get very faithful reproduction of the final furniture piece. So you can see in this Paradox furniture, you have these double-sided screws that don't make any sense. Your furniture is arranged in geometric ways that don't make any sense. And so depending on the quality of your instructions, you're going to get very different outcomes for what you're trying to assemble. And this is very similar for our cells. So our cells have a set of instructions that allow them to be healthy. So our cells have 23 pairs of chromosomes, around 3.2 billion base pairs of DNA. They have their own specific set of epigenetic modifications. And if you're lucky, there's no viruses in your genome either. But in certain cancers, we can have changes in this instruction set for our cells. So in many cancers, we have extra chromosomes or fewer chromosomes. You have a change in the number of your base pairs. You can have a different set of epigenetic modifications, and you can have up to 7 different viruses that can cause cancer. So depending on these input instructions, the output can be either a healthy cell or a cancer cell. So in these cancer cells, many of these changes, the number of chromosomes, the number of base pairs, and the presence of these different viruses often lead to genetic mutations. But conversely, changes in these epigenetic modifications are really a separate topic that's called epigenetics. So what is the difference between these mutations and epigenetic changes? So in very simple terms, mutations are essentially changing the text of our genes. And so this is just an example sentence here of let's eat Fido, a very simple sentence. And in this sentence, you can see that there's this man offering his dog Fido a treat. But if there's a mutation in the text of this sentence, so for instance, if we change eat to play, it dramatically changes the meaning of the sentence. And it changes the meaning of the sentence by changing the actual writing of the sentence. And that's really akin to a mutation from eat to play. However, in epigenetics, we're not actually changing any of the writing in our genes, but what we're still doing is changing the meaning. So in this case, epigenetics are much more like the punctuation of our genes. So this is the same example again, where a man here is offering a treat for his dog Fido, but we can change this punctuation and change the meaning of this sentence. So by adding a question mark, we can turn this statement into a question. Do you want to eat Fido? And again, if you change this sentence by making it just a statement, it really appears that this man is suggesting that he eats his pet instead of feeding his pet. So changes in the punctuation of our genes and the punctuation of our language can have very dramatic impacts on the meaning of these sentences. So what are the components of our cells that can control the punctuation of our genes? So we can take this example sentence again here, where it says, let's eat Fido. And we have components of our cells called readers, and these readers can interpret the meaning of our epigenetic code on these different genes. And in this case, this reader protein is reading this sentence, and it's understanding that this sentence means to eat this pet. But in reality, this is not the meaning we want to have. So this reader will say, this is not correct. And then these reader proteins can recruit proteins called writers that can add new punctuation to our genes or to these sentences. And they can also erase current punctuation. And in doing this, we can change the punctuation of these sentences. And then these reader proteins can read this again and say, this is now asking a question. Shall we eat Fido? And this is, in this case, the correct interpretation of this sentence. But in this case, the meaning has dramatically changed just by changing the punctuation, but not actually changing any of the writing here, which is different from a mutation. So now, ultimately, these epigenetic modifications are not periods, commas, and question marks. What they are is modifications on our genes. And so in this case, in a normal gene, we can have these cancer genes. And if you are a cell and you don't want to become a cancer cell, usually these readers, writers, and erasers will place silencing epigenetic marks on these cancer genes. And that keeps these cancer genes turned off. But if this epigenetic regulation is abnormal, these readers, writers, and erasers can accidentally put activating epigenetic marks on these cancer genes. And these activating epigenetic marks turn on these cancer genes, and this can be a cause for the development of cancer just by changing the identity of these marks on these genes without ever changing any of the genetic code of these genes. So obviously in our genome, we don't have boxes that signify our genes. So in reality, what our genes look like is this, where we have, obviously there's DNA, and then there are these proteins that our DNA gets wrapped around called histones. And the complex of this DNA and this histone is called the nucleosome. And these epigenetic marks can be placed either directly onto our DNA, so modifying our DNA, or they can be placed on these histone proteins that the DNA gets wrapped around. And all of these modifications are similar in that they are all chemical modifications of either our DNA or of these histone proteins. And if we now look at the atomic model of the nucleosome, so this is an actual atomic model of this nucleosome where the DNA is wrapped around histones. And in this case, every little ball represents an atom in the nucleosome. So if we look at this atomic model of the nucleosome, we can see that these epigenetic modifications can have a variety of shapes and sizes. So in the most extreme case, we can have modifications called Ubiquitin. And Ubiquitin is a very large modification of almost 3,700 atoms. It's very chemically complex. It encodes all these different elements that are bonded in many different ways. And because it's so large and it has such complex chemistry, it can interact with many different epigenetic readers, writers, and erasers. But there are also different modifications. And you may have missed it, but there is now another epigenetic modification on this nucleosome. And you might have missed this because this modification is very small, and it's called lysine methylation. And so compared to ubiquitination, lysine methylation is very, very, very small. It is only around 4 to 12 atoms in size, depending on which state it's in. It's very chemically simple. It's only composed of carbon and hydrogen, and there's no interesting new bonding going on. And because it's so very small, it tends to only interact with a very specific set of readers, writers, and eraser proteins. So this really illustrates the huge chemical complexity that can be attached in these epigenetic modifications. So who are the readers, writers, and erasers that are responsible for changing this epigenetic modifications and epigenetic content on our genes? Ultimately, all of these readers and writers and erasers are proteins. They're proteins within our cells that operate as a machine. And my lab is very interested in trying to understand how these different protein machines can affect our epigenome. And the way we want to do this is can be inspired by Richard Feynman, where he said, if you want to answer these fundamental biological questions, you can just look at the thing. And so what we do in my lab is we actually look at these protein machines. And the way we do that is through this process called cryo-electron microscopy. And so this is an image from Big Bang Theory of Sheldon looking through a typical light microscope. And so this is probably what most people think of when they think of microscopy, these small benchtop light microscopes. But because we are not using light, we use electrons for our microscopes, we have to use very large microscopes. So this is a picture of me in our cryo-EM facility here at the Van Andel Institute. And this big white and gray box to the right of me is our microscope. And these microscopes are really very large, around 11 feet tall. And the sample we put into these microscopes is very tiny. So I'm actually holding in my hand a cryo-EM sample. And this sample is a small metal disc, and it's so small you have to hold it at the end of tweezers. And we call these grids. And these grids are called grids because if you zoom in on this sample, you can see that it is an array of grids. And you can see in this grid pattern, there are some holes that are large and clear. There are other holes that are very small. And there's other parts of this grid that are completely opaque. And the reason for that is that in the microscope, these grids are covered with an incredibly thin sheet of ice. And this thin sheet of ice is filled with a protein that we're interested in studying. So if we zoom in one more time onto a grid square, we can see that these grid squares have an array of holes. And if we zoom in one more time on one of these holes, we can see that there are proteins suspended in this ultra-thin layer of ice. And each of those dots in this image here is an individual protein that has been purified and frozen and put onto our electron microscope. And then once we collect millions of these individual particle images, we can reconstruct the three-dimensional shape of these proteins, which we can then use to understand fundamental mechanisms about how these proteins function. So one of the particular proteins we are interested in is called DOT1L. And so DOT1L is a reader and writer protein that has a catalytic domain, that box in gray. And it also has a long unstructured portion of the protein that has small pieces that interact with other reader proteins. And if we look at the structure of these two components, we can see that the catalytic domain has this well-folded structure. And so this set of helices and these arrows are showing a simplified version of the protein structure. And if you have a well-folded protein structure, you'll see a lot of these helices and beta sheets is what they're called, indicating that it's a well-folded protein. And the catalytic domain has two activities. It can be a reader protein, and it can also be a writer protein. And this long extension that interacts with other reader proteins is not well structured. And you can see that through this model that really shows it as just a bundle of spaghetti. And the only parts of this protein in this unstructured region that have any structure at all are these parts that interact with these different reader proteins. And so when we think about DOT1L, we can actually think of it like two of these different people, right? One person is DOT1L, that's a reader and a writer, so he has a magnifying glass and a pencil, and he's tethered to another reader through this leash, which is representing this long unstructured region. And so this pairing between the DOT1L catalytic domain and a reader protein allows this reader protein to tell DOT1L where to go to attach these histone modifications. And so how it works in our cell is that, in a normal cell, this reader protein will tell DOT1L to go to this good gene. He says, go there. And then DOT1L will turn on this gene by adding activating histone modifications to this good gene. And this is what you want in a normal cell. You want these good genes to be activated. However, in certain cancers, these reader proteins can actually be mutated. And when these reader proteins get mutated, they don't tell DOT1L to go to the good genes. These reader proteins then direct DOT1L to go to a cancer gene. And DOT1L can't tell a difference here. It requires this reader to tell it where to go. So when the reader tells DOT1L to go to these cancer genes, DOT1L very dutifully then attaches these activating histone marks to these cancer genes. And so these mutated reader proteins can direct DOT1L to these different parts of our genome, and then DOT1L will activate these cancer genes by attaching these activating histone modifications there. And there's another lab here at the Van Andel Institute, the Hong Wen lab, who's very interested in these mutated reader proteins. But I was very interested in particular this protein DOT1L, and I wanted to understand, how does DOT1L actually attach these activating epigenetic modifications to these genes? And to understand DOT1L's activity, we need to understand how it has this reader function and writer function. So this is an atomic model of a nucleosome again, and we see there's a ubiquitin modification on the nucleosome, and there's also a lysine modification on the nucleosome. So DOT1L has two specific activities that first allow it to recognize and read this ubiquitin modification. And if DOT1L sees this ubiquitin modification, that then allows DOT1L to write this lysine methylation. And if there's no ubiquitin here, DOT1L cannot see the ubiquitin, obviously. And then if it can't see the ubiquitin, it also can't write this lysine methylation. So in this case, the activity of DOT1L to write this lysine methylation is strictly dependent on the presence of this ubiquitin modification. And so I was very interested in trying to understand how DOT1L is able to read this ubiquitin mark and then at the same time write this lysine methylation. I really wanted to understand the connection between these two histone modifications. And to do that, I used cryo-EM to determine a structure of the DOT1L catalytic domain bound to a ubiquitinated nucleosome. So this is the experimental result of cryo-electron microscopy. What you get at the end of an experiment is a three-dimensional density, is what it's called. And you can see in this density different features of your complex. So we can clearly see in gray, there's this double helix of DNA wrapping around the nucleosome. In blue and orange and red and cyan, we can see these are the different histone proteins. And in green, we can see that there's DOT1L, the protein that I'm interested in. And in purple, we can see ubiquitin. So by looking at the three-dimensional density of these complexes you get from the cryo-EM experiment, you can see how these different components fit together. And by looking how they all fit together, you can understand how these proteins then are able to communicate with other components or with these histone modifications. So in this case, when we look at the interaction between DOT1L and ubiquitin, you can see that DOT1L directly binds to ubiquitin. So this is how it reads and recognizes this ubiquitin modification. And if we take this portion of DOT1L that interacts with ubiquitin and we mutate it targetedly, we can actually reduce the reading activity of DOT1L. So in this bar graph, the wild type bar shows the full activity of just wild type DOT1L. But when we mutate this interaction site with ubiquitin, the reading activity of DOT1L drops by 80%. So this validates that this portion of DOT1L that we see binding to ubiquitin in the structure is actually the part that is responsible for letting DOT1L read this ubiquitin modification. And in the structure, we can also see that DOT1L makes contacts with the histones in the nucleosome that are right around its substrate lysine. And so I determined also a second structure of this complex, but this other structure was an inactive structure, or a structure that we termed a poised structure. And in this other structure of DOT1L, and I'm showing this as this ribbon representation, which is just a simplified way to look at a structure. We can see in this structure that the DOT1L catalytic domain is no longer touching these other histones in the nucleosome. It's completely lifted up off the surface of the nucleosome. And really critically, there are these catalytic loops that are very important for the function of DOT1L that are completely disordered. And if we compare this poised state with the active state, we can see that when it moves down to the active state, it's really pivoting around this ubiquitin modification. So it looks like this ubiquitin modification is serving as a rotation point for DOT1L to switch between the inactive poised state and the active state. And if we look at this active state structure, we can see that these catalytic loops that were completely disordered in the inactive state become structured, and we can see where they are. And we can also see that DOT1L is making specific contacts with the lysine that it's going to methylate. So there are other motions going on when DOT1L transitions to the active state. And these motions occur in the histone residue that DOT1L wants to methylate. This is called H3 lysine 79. So in the inactive state, where DOT1L is not touching the surface of the nucleosome, this lysine residue is tucked in a little groove. And being tucked in this little groove actually stops this lysine from being accessible for modification. And what happens when DOT1L transitions to the active state is that this lysine gets lifted up and pulled up out of the surface of the nucleosome, and it moves by 10 angstroms to point straight into the air here. But it's not actually in the air, it is inside the DOT1L active site. And the outcome of this movement of this lysine side chain is that this lysine then becomes very close to this important cofactor for DOT1L that donates the methyl group for the lysine methylation reaction. And then in this case, this methyl donor that's on this cofactor called SAM goes within 3 angstroms of the lysine side chain that's going to be methylated. And this very close distance between this lysine side chain and this SAM methyl donor will allow this lysine to be methylated by this SAM cofactor. And so this really answered how DOT1L is able to access its substrate lysine and how that then allows it to get methylated. So in summary, this structure of DOT1L bound to a nucleosome revealed several interesting features of the activity of DOT1L. So first, we figured out how DOT1L is able to read this ubiquitin modification. It actually physically binds to the ubiquitin. And binding to that ubiquitin allows DOT1L to pivot around the ubiquitin. And we think that this ubiquitin really restricts the movement of DOT1L, so it can only switch between 2 states, active and poised or inhibited. But if there was no ubiquitin there, DOT1L may still be able to bind, but it might just move very, very far, all over the place. And that's been shown by other groups that when there's no ubiquitin, this DOT1L can just move everywhere. And so restricting its movement between only these 2 states, we think allows it to more frequently access this substrate lysine. And being able to access this substrate lysine allows then this writing activity of DOT1L to attach a methyl group to the end of that lysine, which is the writing activity. And I didn't mention it, but that we also in this structure identified a second reading activity that wasn't previously known, where DOT1L can read this other part of the nucleosome called the H4 tail. But yeah, with that, I would be happy to take any questions.
Great. Thank you so much for that very, very deep dive and that wonderful presentation. A couple questions have come through, and there's always one we'd love to know. What led you to a field of cancer research? What was the spark back in the day? Was there a spark? Why are you doing what you do now?
Yeah, I would say I sort of fell into cancer research because I really first got hooked on structural biology, which is the study of these protein structures. And because cancer interacts with so many fundamental systems in our cells, there are a lot of fundamental questions about cancer that can only be answered with structure. So my first love was really doing this structural biology, but then there were just so many questions in cancer research that are answerable by structural biology. I really got into that from structural biology first.
Nice. Thank you. Well, an extremely technical question has come in, so I'm sure you can answer it. But how many lysine methylation particles are available in comparison to ubiquitin markers in a nucleosome that the ubiquitin can impact them and use the .1L to initiate its activities? That was a mouthful, but I punt that over to you.
Okay, so the specificity of .1L is very strong. So there's only one single ubiquitin modification that can stimulate its activity. However, there are other proteins that interact with that specific ubiquitin modification that write other lysine methylation marks on the nucleosome. And in addition to that, there are many writer proteins that can write lysine methylation that do not depend on ubiquitin at all. So there's all these different epigenetic combinations, and some of them depend on ubiquitin, some of them depend on other marks. But I think in general, there are at least 8 or 9 lysine methylation sites that are very important that people study a lot. And there are at least 4 different ubiquitination sites on the histones that can control other epigenetic modifications. But for .1L, it really only reads that one ubiquitin mark, and it really only modifies that one lysine. Yeah.
Okay, good. A couple more questions have come in. That's great. What makes this process different for slow versus fast-growing carcinomas?
Yeah, I'm not sure how to answer that. So I don't, because I'm studying structure primarily, I don't usually get involved with the cell biology of cancer. I do know that these .1L mutations, right, these reader proteins that direct .1L to different sites, are primarily associated with blood cancers, like mixed lineage leukemia is the primary one. And that was the cancer type that was actually informative to know that dysregulation of .1L was critical to formation of those cancers. But I'm not sure I have the capability to answer that specific question.
Okay, no problem there. Tell us more about the role of ubiquitin in cancer, and also what are some of the implications of your research?
Wow, ubiquitin in cancer. So the ubiquitin field is extremely large. So we're studying a very, very tiny part of ubiquitination, which is these monoubiquitinations on chromatin, on these histone proteins. But ubiquitin in general is very tightly linked to cancer because it's so huge. So ubiquitin can be a single ubiquitin on a protein like a histone, but you can also form these long ubiquitin chains. And these chains can have different structures and geometries. And the huge number of these different chains makes it possible to have many different proteins that interact with these ubiquitin chains. So there are other processes completely unrelated to chromatin and to epigenetics that are involved with ubiquitin that are important for cancer. One of the primary ones is protein degradation. And in that case, protein degradation needs one of these ubiquitin chains for a protein to get destroyed when it needs to. And the process that happens is something called it gets directed to something called the proteasome. And the proteasome is a cancer target. And so there are already several anti-cancer medications that inhibit this proteasome, which is the end product of this very complicated ubiquitin signaling. So ubiquitin has a much bigger role in cancer than its role in chromatin, but its role in chromatin is also very important for cancer.
Great, thank you. Another question has come in. Are all cancers caused by methylated lysine?
No, there are many cancers that are caused by genetic mutations. And those are going to change the genetic code of a certain gene to make it defective in some way. And those cancers aren't necessarily going to have a change in their epigenetic modifications, but they will just change the function of the protein. Yeah.
Perfect. What are you most excited to see in the future of cancer research?
Yeah, so again, the cancer field is so large. I feel I couldn't adequately cover, generally in the cancer field, but in the structural biology field, especially as it relates to cancer, I'm most excited about new techniques that people are using to determine these protein structures. There are new cryo-EM tools that we can use to, instead of, taking an isolated protein and collecting millions of images of that isolated protein, you can take a cancer cell, for instance, and slice the cancer cell very, very thinly. And then you can look at all these proteins in the cell, and you can understand how these proteins are interacting with different organelles in the cell or other different proteins in the cell. And that's called cryotomography.
Okay.
I'm also very interested and excited about these new structure prediction tools called AlphaFold or Rosetta Fold. And these protein prediction algorithms can allow you to get an idea of what a protein looks like without having to do the EM experiment. And it is quite accurate. So I'm very excited about those two things. And we're in a little bit of a golden age of structural biology. And I'm most excited about those two things.
Fantastic. And one last question, you kind of hinted at this in your last answer, but what do you consider, what's the biggest gap today in knowledge in cancer research? What's that gap that we hope to fill and to get rid of at some point, hopefully?
Yeah. So I think in terms of the epigenetic connection to cancer, I think there are several unknowns about how these epigenetic marks actually influence cancer. We mostly understand associations between an epigenetic mark and gene activation, or an epigenetic mark and a cancer type. But it's not always clear, or I would say it's seldom clear, exactly what that different epigenetic mark or the different location in the genome is doing. Because either we don't know the function of the epigenetic marks at all, or we don't know the reader proteins for these epigenetic marks, or because we just don't really know how everything comes together to turn a gene on or off epigenetically. And so I think the big unanswered questions really revolve around, the function of these different epigenetic marks that we see in cancer. And those questions of function are really what most people are working on. And it's still a very big unanswered question because there are over 100 different epigenetic modifications and there's different combinations of them. So really understanding the role of these different epigenetic modifications in cancer generally is a big unanswered question.
Sure. Another question has come in. This is great. Do you feel there will be, or are there nutritional treatments that will help cancer care to a positive end?
Oh my goodness. Yeah, I'm not sure I have the ability to answer that. I don't really know much about nutrition in cancer. I know there are some people who have found that nutrition can, for instance, change some epigenetic modifications. But in the more general sense, no, I don't really know how nutrition will affect cancer. Although there is a metabolism department here at the Van Andel Institute that directly studies those types of questions.
Fantastic. I don't see any other questions coming in. So thank you everyone for joining us today. Dr. Worden, any parting thoughts before we sign off here today?
No, just thank you for giving me the opportunity to present my work, and I'm really happy to be here and share my science with the broader community.
We actually have a question that just came in. All right. So what methods have you learned that can slow or block the interaction between the .1Ls and the lysine to prevent tumor development?
Right. So there are several targets, right? So a lot of people are very interested in these mutated reader proteins because that's really the part that goes wrong in these cancers, right? These mutated reader proteins take .1L to the wrong place. And so those mutated reader proteins are a potential area that you could drug a cancer cell. So if you could figure out a way to specifically target these mutated reader proteins and stop them from taking .1L to the wrong place, that's a potential treatment that people are pursuing. There's also inhibitors of .1L. Say you can't actually stop targeting .1L to these cancer genes. Maybe you can just stop .1L from writing these epigenetic modifications. And so there's a few companies who have started making these .1L inhibitors that stop its writing activity with the idea that maybe that can stop these cancer genes from being turned on. However, and unfortunately, in clinical trials, these DOT1L inhibitors have not really been effective, as far as I can tell. And that means maybe the whole process is more complicated because there are other proteins that get recruited abnormally in these cancers. And so it could be a combination of factors that lead to cancer formation. But they're really focusing on these reader proteins and the DOT1L activity. That's what people are usually targeting for cancer treatment.
Fantastic. Thank you. Yeah, we've had a total of 9 questions that have come in today. That's outstanding. So, Dr. Worden, thank you very much for your presentation. I'd like to thank everybody online for taking the time out of your schedule to join us today. Please stay active and engaged with Van Andel Institute. You can find out more information about us at vai.org. We will have many more public lectures and other events in 2023, but thank you once again for attending. Have a wonderful afternoon and have a fantastic holiday season. Thank you.